Hi everyone, good evening. This is going to be the portion of the notes from the professor on the enzymes that are derived from microbes. Also, the test is going to be on the 2nd, April 2nd. Alright, history. So, 1894, there's a fungal amylase that was being used for digestive disorder medication in the pharmaceutical agency. And in 1915, that's when we see these tryptic enzymes. These are going to be found in laundry detergents. By 1969, 80% of these detergents will have enzymes. The allergies associated with the enzymes in the detergents cause a drop in the usage of these microbe enzymes until we had these micro encapsulated enzymes as well as the liquid detergents. Then we don't have to use this solid powder enzyme, we just use a liquid detergent, so then you use them again. In Tide, you have 4 enzymes. These are all derived from microbes. You have a protease, you also have an amylase, you have a mannase, and a pectinase. Now, combining all the forces of these three, so for example, protease works on proteins, amylase will work on starch, mannase again works on a carbohydrate, then pectinase, you're able to basically get rid of most stains, which is very nice. Usually the enzymes are going to be made in very low concentrations in wild-type organisms. So, in order to bolster or boost the amount of enzymes that the organisms are actually going to be making, you could use these different techniques. You could edit the genetics of the organism. You could also optimize the medium and fermentation. That's what we're doing actually in the lab portion, that we are optimizing the M63 for the E. coli biomass. Well, in the case of the enzyme producing microbes, you could use a low-cost feed, and you could also use fed batch fermentation. The release of enzymes, so the amount of enzymes that the cells are going to be releasing, you could increase that by cell breakage techniques. You could also change the way you're purifying the product, so you could have ion exchange and affinity chromatography to increase that concentration and the purification of your product. You could also immobilize the enzyme. So you're going to put those enzymes onto this medium that they bind specifically to, and then you're just going to pour whatever molecule you need transformed by the enzyme down this gradient or this medium that has the enzymes conjoined to it in a way, or bonded to it, and then this will decrease the amount of enzyme needed to perform that task or that transformation of whatever molecule you're trying to transform using the harvested enzymes. And then you also have continuous production. You would need smaller size equipment for that. Okay, amylase. So this is going to be the enzyme for starch. Starch is a plant sugar. It's going to be used for storage. And an amylase is a hydrolysis enzyme. The main use of the amylase would be in the food industry, and you're going to use it for sweeteners. So a basic overview of the process: first, starch is converted to short-chain polymers known as dextrins, and then the dextrins are converted to maltose. Maltose is a disaccharide, and afterwards you get to maltose, you go and convert that to glucose. So we have several enzymes here that work on starch that we need to take into account. We have alpha glucosidase. A glucosidase will split the glucose from the non-reducing ends to yield these free glucose. We also have a limit dextrinase known as a pulinase, and this will break alpha 1,6 bonds. So pulinase will break alpha 6,1 or alpha 1,6 bonds. Sorry about that. So in Starch, you have these branches. So remember the alpha 1 4, those are going to be just the glucose in like a straight path, if you will. There's a branching point with this alpha 1 6 bond, and then that creates a branch, and it just makes the molecule more complex. The polynase works on the 1 6 glycosidic bonds. Next enzyme is the alpha amylase. So this is the amylase itself, a type of amylase. This will break the alpha 1 4, and you're going to cut these alpha 1 4 bonds to make these oligomers. However, on the beta amylase, this is different. You're going to cut the alternating alpha 1 4 bonds. And this is going to make a disaccharide. This is going to make maltose. Maltose is 2 glucose together. So beta, you're going to alternate which of those alpha 1 4s you're going to cut and make a disaccharide in maltose. But alpha amylase will just cut the alpha 1 4s to make oligomers. Let's talk about high fructose corn syrup. This is inexpensive. You could compete with the sucrose industry in the production of high fructose corn syrup. The relative sweetness is very interesting. So glucose is 75% sweeter than sucrose. However, fructose is 2 times as sweet as sucrose at half the weight. So that's very interesting. Fructose is extremely sweet tasting. Alright, step 1 to make high fructose corn syrup would be liquefaction. This is where you're going to take the starch and you're going to convert it to low dextrose equivalent maltodextrin. So these are going to be low dextrose equivalent. Okay, so with the monosaccharides and the disaccharides, you have a very small percentage. Like, for example, glucose is only 1 to 20%. However, in the higher oligomers of these sugars, you're going to have 50 to 80%. So there's more oligomers made instead of just like individual units or disaccharides. Okay, conditions for this: neutral pH at 7, 95 degrees Celsius to 107 degrees, and that's going to be a raise. You're going to raise the temperature from 95 degrees to 107 degrees Celsius. You could also use 20 parts per million. Calcium to stabilize enzymes. You could use the calcium to stabilize the enzymes in liquefaction. Right, we're going to have our alpha amylase from Bacillus subtilis, and that's going to cause a paste to form. Okay, now saccharification. You're going to take the low dextrose equivalent maltodextrin, and you're going to convert it to glucose. You're going to need 2 enzymes for this. You need glucoamylase from Aspergillus niger, and this is going to split the alpha 1 4 linkages to release glucose in a stepwise process from the reducing end of the starch chains. That's the glucoamylase. Polynase, we've seen this one before. You get it from Bacillus. It splits alpha 1 6 linkages. You need these other conditions, though, for step 2 in the saccharification. You need pH 4 to 5, so slightly more acidic. You also need to lower the temperature quite a bit. They're going to be at 55 to 60 degrees Celsius. This prevents the formation of colored products, aka calcium. I mean, not calcium. Sorry about that, aka caramel. Sorry about that. When we're talking about colored products here in saccharification, if you raise the heat too high, you get caramel. Not calcium, caramel. Alright, third step is isomerization. This is originally carried out in batch with stirred tank reactors. However, now we just use the immobilized enzyme reactor according to the notes of the professor. The column will have this glucose isomerase kind of immobilized or embedded into it. And now the enzyme, since it's bound to the solid support by covalent and no covalent bonding, now again you use less enzyme. You're not washing it off. Okay, so you add the stuff from bottom to top to stop air, and 1 kilogram of the enzyme will make 18 tons of the high fructose corn syrup. Very important to understand this process. The reaction for isomerization is carried out at 42% fructose. And you're going to have increasing concentrations of this by using cation exchange chromatography. Fructose, again, is more strongly absorbed than glucose, and the fructose is, again, 2 times sweet as sucrose. Alright, sorry about that. Okay, you're going to be using, again, glucose isomerase, so it's actually D-glucose ketoisomerase. And, again, glucose and fructose are isomers of each other. Right, let's talk about the application now of A amylase. So we talked about the product, the high fructose corn syrup. Let's talk about the enzyme that is harvested from the microbes. Okay, so how do we use A amylase or alpha amylase? In milling, we're going to modify the amylase deficient flour. In starch, you're going to have the liquefaction up here, step 1. In high fructose corn syrup, you need the alpha amylase to make this paste. Okay, baked goods, you're going to increase the amount of fermentable sugars in them. Paper, again, liquefaction, but you're going to have the liquefaction of starch without sugar production for sizing. Feed industry, you could improve utilization by treating barley. It's easier to digest for the animals. And sugar, you're going to increase the filterability of the cane juice by breaking down starch and juice using this alpha amylase. And then laundry detergent. Okay, let's talk about the microorganism that makes our alpha amylase. This is Aspergillus oryzae or Bacillus subtilis. So the stock now will be all about 5% starch, peptone yeast extract, ammonium nitrate, so, the normal players here that you would expect, and minerals. Okay, now going further into Bacillus subtilis, now talking about the process in the fermenter. This is going to be batch or fed batch. It's most made during, so the enzyme itself, it's made most right before sporing. Before sporulation, you're going to see an increase in amylase production. However, the rate of formation is low during exponential phase. You have this rating here, the fermentation conditions for Bacillus subtilis, 27 to 30 degrees, pH 6.8. Next, you're going to just centrifuge and ion exchange to continue processing and purifying your amylase that you isolated from Aspergillus oryzae or Bacillus subtilis. Okay, how do we stabilize purified enzymes? And this is very important because enzymes are unstable, so they have this limit on them. They have a shelf life. Okay, and they could be stabilized by accomplishing the addition of a chemical, so like an additive treatment. That's for soluble enzymes. You could also do cross-linkage, and you're going to cross-link enzyme molecules together, but this does not change affinity. It doesn't change the affinity when you do cross-linkage to stabilize the enzyme. Okay, you also have the enzyme is just bound to a carrier instead of its other, you know, brothers or sisters in the enzyme mix. It's just bound to a carrier that isn't the same enzyme. You also have incorporation of semi-permeable membranes. So these include the enclosing of enzymes into gels, microcapsules, and these fibrous polymers. The pores are small enough so the enzyme doesn't escape. However, the pores are large enough for the substrate to enter. So again, the Goldilocks effect is at play. All right, thank you so much for tuning in. Please do something nice for someone. Best of luck.